Welcome to Section 2 of the Renal Physiology Review. In Section 2, we will walk through renal functional anatomy, introducing the renal functional unit, or the nephron. Welcome to Section 2 of the Renal Physiology Review. In Section 2, we will go through an overview of renal functional anatomy. As mentioned in Section 1, much of the material will be repeated. The purpose of the repetition is to provide you with a better understanding of renal functional anatomy so that we may take this, build upon it, to move to renal physiology. This is an overview of the renal functional anatomy. As we said in Section 1, the functional units of the kidney is the glomerulus. This slide provides an overview of the renal functional unit with the glomerulus and its associated tubular structures. Once the blood passes through the glomerulus and forms a filtrate, the urine passes through Bowman's space into the proximal convoluted tubule. After the proximal tubule, the next segment is the thin descending limb of the loop of Henle. It then passes into the thick ascending limb of the loop of Henle. Proceeding forward, it goes to the distal convoluted tubule, finally into the collecting duct, first the cortical, then the medullary collecting duct. We will highlight each of these segments individually, but this slide merely provides a brief overview. There are two main regions of the kidney, the cortex and the medulla. The cortex comprises 75% of the kidney mass, the medulla about 20%. The medulla is composed of the renal pyramids, the renal columns, the calyces, and ultimately the renal pelvis. Important to note here, the cortex includes most of the renal glomeruli. Therefore, when kidney biopsies are taken and pathologic diagnosis must be made of glomerular disease, cortical specimens must be sampled. Also important to note is that the renal medulla, its deep location within the kidney, makes its environment have one of the lowest PO2s in the body. The nephron is considered the functional unit of the kidney. Most individuals are born with 1.2 million nephrons per kidney. There are three major types of nephrons. There are superficial cortical nephrons, which include a short loop of Henle and only reach the outer medulla. Mid-cortical nephrons, they can have short or long loops of Henle. And finally, juxtamedullary nephrons. These have large glomeruli and long loops of Henle, which extend into the inner medulla and the papillae. The importance of the juxtamedullary nephrons is that they provide a large surface area for urinary concentration, which we will highlight in a later segment of this review. Nephrons are composed of the glomerulus. As mentioned before, once the blood passes through the glomerulus and the filtrate is made, the urine then passes through the proximal convoluted tubule. It then proceeds through the loop of Henle, first the thin descending limb, and then the thick ascending limb, then into the distal convoluted tubule. Finally, it enters the collecting duct, first the cortical collecting duct, and the medullary collecting duct. Each segment has a unique cellular makeup, specialized for its particular role in the control of fluid-electrolyte balance, again, which we will outline in a later portion of this review. This is another view of the renal functional unit, or the nephron. Once again, you see the glomerulus and the glomerular capillaries, Bowman's space. The urine then passes into the proximal convoluted tubule, the proximal straight tubule, the thin descending limb, the thin ascending limb, the thick ascending limb, the distal convoluted tubule, the cortical collecting duct, the outer medullary collecting duct, and then the inner medullary collecting duct. Highlighted in this slide is the sample known as the macula densa. Importantly, the macula densa is between the thick ascending limb and the distal convoluted tubule. The macula densa has a direct communication back to the renal microcirculation. The macula densa plays a role in the regulation of the glomerular blood flow, filtration fraction, to once again regulate the final urinary composition. Renal blood flow. We've outlined the macrocirculation of the renal blood flow. However, this is the microcirculation. It starts with the afferent arteriole. The afferent arteriole brings whole blood to the glomerulus of each nephron. Once the blood enters the glomerulus through the afferent arteriole, the filtrate is formed. The blood leaving the glomerulus exits through the efferent arteriole. This takes concentrated or plasma-reduced blood from the glomerulus to the peritubular capillaries and the vasa recta. The vasa recta contains blood which is plasma-reduced with increased concentration of proteins, blood cells, and glucose. The purpose of the vasa recta is to serve as the partner circulation to the renal tubules, to provide a site for reabsorption of important electrolytes, glucose, amino acids, as well as a potential source to secrete unwanted substances or medications into the tubular lumen. It is extremely important to remember that the afferent arteriole enters the glomerulus and the efferent arteriole exits the glomerulus. One may find it easy to remember to simply think this is alphabetical. A comes before E in the alphabet, and A comes before E in the renal circulation. Once again, this is the functional unit of the kidney. We will highlight this again. The afferent arteriole enters the glomerulus, the filtrate leaves, goes through into the proximal convoluted tubule, into the loop of Henle, the distal convoluted tubule, and the collecting duct. This slide, though, better highlights the interconnected vasa recta with the renal tubules. Once again, when the substances have been reabsorbed or secreted, the final blood then leaves into the venules and into the renal vein. Let's go through a question. During an autopsy, organs are routinely weighed to detect possible atrophy or hypertrophy. On a per-weight basis, which of the following organs receives the largest percentage of cardiac output? A, the brain, B, the kidneys, C, muscle, D, heart, or E, the skin. This being a renal physiology course, the answer is clearly B, kidneys. The two kidneys have a combined weight of roughly 300 grams, yet they receive about 20% of the cardiac output in each cardiac cycle. This corresponds to a rate of 600 mL per minute for each kidney. It is not surprising then the renal artery has the highest systolic pressure of any other organ bed, and also makes it easy to understand why the kidneys are so susceptible to variations in blood flow or blood pressure. Now that we've outlined the renal functional unit anatomy, or the nephron, let's go through some basic renal physiology. In making the final composition of urine, there are two basic processes. First is filtration. Glomerular filtration is selective based on both a size as well as a charge barrier. This is the first process in the making of urine. The filtration is selective based on size and charge, but is not selective in terms of how much salt and water is filtered. This must occur through modification of filtrate. Modification occurs through two major processes. One, reabsorption. Two, secretion. The reabsorption and secretion occurs throughout the renal tubules. Depending on the substance and the blood concentration of the substance, how much of that substance is reabsorbed or secreted can be modified. Renal filtration occurs at the renal glomerulus. This is an image of glomerular anatomy, including Bowman's capsule. As we've said before, blood enters the afferent arteriole, enters the renal glomerulus, goes through the filtration process, and the urine then enters Bowman's capsule. Blood then leaves the afferent arteriole. You can see here once again the macula densa and its relationship to the afferent arteriole. This will become important when we discuss the regulation of afferent arteriole blood flow and how that affects glomerular filtration rate. As we've said, filtration occurs in the glomerulus. Blood enters through the afferent arterioles into the glomerulus and expands into a tuft of capillaries. The tufts increase the surface area of the capillaries dramatically, which greatly enhances the amount of filtration possible. This is an important thing to remember, is that the surface area of the capillaries can expand or contract depending on the activity of the mesangial cells. This is another way the kidney can regulate glomerular filtration rate, which ultimately regulates the final urine product. Filtration occurs in the capillaries. The capillary tufts are fenestrated. The fenestrations allow fluid to be filtered into the glomerulus while preventing large particles such as proteins and blood cells from passing through the barrier into Bowman space and into the tubular lumen into the urine. The filtration barrier is made up of three important layers. First is the blood side, with fenestrated endothelial cells on the glomerular capillaries that serve as the initial barrier to the prevention of large proteins and blood cells from passing through. Secondly is the glomerular basement membrane. The glomerular basement membrane is negatively charged, which repels negatively charged proteins. The most abundant negatively charged protein it needs to repel is albumin. Finally, on the urinary side, there are podocytes, which are known also as visceral epithelial cells. They lie over the glomerular basement membrane. Between the podocytes are tiny openings or filtration slits, which are covered by slit diaphragms. It's important to remember that pathology at any of these sites, either on the blood side with inflamed or damaged capillaries, damage to the basement membrane, or damage to the podocytes, causes this barrier function to be impaired and may allow things unwanted, such as red blood cells and proteins, to pass into the urine. What is the primary protein found in the serum? As we said, albumin. And what is its charge? Negative, just like the glomerular basement membrane. This is an example of how the specialized characteristics of each of the three parts of the barrier provide an additional layer of protection to prevent unwanted substances, proteins, blood cells, etc., from passing into the urinary filtrate. Let's digress into a clinical correlation. Can you think of a condition in which the negative charges on the basement membrane are lost, resulting in proteinuria? The condition is known as minimal change disease. Minimal change disease is a type of primary glomerular disease in which the glomeruli appear normal on light microscopy, hence the name minimal change. For unclear reasons, negative charges on the glomerular filtration barrier are lost. Proteins, particularly albumin, are able to pass through the basement membrane, resulting in severe proteinuria. The etiology for the vast majority is idiopathic. In adults, some cases are drug-induced, non-steroidal drugs are a common culprit, or paraneoplastic syndromes, most commonly Hodgkin's lymphoma. Clinically, this is the prototype of the nephrotic syndrome, and is the most common cause of nephrotic syndrome in children. In adults, however, it only accounts for approximately 10% of nephrotic syndrome patients. The diagnosis of minimal change disease is often clinical in children. In children with nephrotic syndrome without another underlying disease process, biopsy is generally not needed, and these patients are treated empirically with corticosteroids. However, in adults, a biopsy is required due to a broader differential diagnosis. The disease is named, as are many of the primary glomerular diseases, for its biopsy findings. The biopsy findings are normal on light microscopy. However, on electron microscopy, we see effacement or fusion of the foot processes. As I mentioned, the treatment is corticosteroids. In children, this usually results in a dramatic improvement. The failure to respond to treatment suggests an alternative diagnosis, such as focal segmental glomerular sclerosis, or other primary glomerular diseases. This concludes Section 2 of the Renal Physiology Review. The key components of Section 2 to remember are that the renal functional unit is the nephron. Once blood passes through the glomerulus and the ultrafiltrate is formed, within the nephron, through secretion and reabsorption, the final urinary composition is determined.